The earliest historical evidence of dengue-like illness were found in China in 992 Anno Domini. They described the disease as water poison and noted it to be associated with flying insects. The first account of a disease resembling symptoms of dengue was reported in Philadelphia in 1780. In the year 1906, Thomas Lane Bancroft suggested that the mosquito vector Aedes aegypti transmitted dengue. The following year, in 1907, Ashburn and Craig proved that dengue was caused by a virus. Japanese scientists Ran Kimura and Susumu Hota were first to isolate the dengue virus. These two scientists were studying blood samples of patients taken during the 1943 dengue epidemic in Nagasaki, Japan. A year later, Albert B. Sabin and Walter Schlesinger independently isolated the dengue virus. Both pairs of scientists had isolated the same virus that is now referred to as dengue virus 1. The first documented cases of the severe form of dengue were seen in Philippines and Thailand during the 1950s. This severe form of dengue subsequently spread throughout Southeast Asia and expanded globally. In 2012, dengue was classified as the most important mosquito-borne viral disease in the world by the World Health Organization (WHO). Dengue is also the world's most rapidly spreading and geographically widespread mosquito-borne disease. The global incidence of dengue had grown dramatically, with about half of the world's population now at risk. Although an estimated 100 to 400 million infections occur each year, over 80% are generally mild and asymptomatic.
Let's dive into the introduction to dengue virus and its pathogenicity. The dengue virus is a single-stranded positive sense RNA virus belonging to the Flaviviridae and is transmitted to humans and other primates by Aedes mosquitoes, importantly Aedes aegypti. The dengue virus has a roughly spherical shape with a diameter of 50 nanometer. The nucleocapsid, a structure made up of the viral DNA and C proteins, is the viral nucleus. The viral envelope, a membrane made up of a lipid bilayer that is obtained from the host, is present around the nucleocapsid. 180 copies of the E and M proteins that cross the lipid bilayer are integrated into the viral envelope. The dengue virus 1, dengue virus 2, dengue virus 3, and dengue virus 4 are four closely related viruses that cause dengue illness. This is because each of these four viruses interacts differently with the antibodies in human blood serum. They are referred to as serotypes. Although the four dengue viruses are related and share about 65% of their genomes, there is genetic diversity even within a single serotype. Despite these differences, each dengue serotype infection causes the same sickness and range of clinical signs. There are three types of transmission of dengue. The first one is through mosquito bites by Aedes aegypti mosquito. Mosquitoes can become infected when they bite a person infected with the virus. So, infected mosquitoes can then spread the virus to other people through bites. Next, dengue can be transmitted from a mother to a child. A pregnant woman who is already infected with dengue can pass the virus to her fetus during pregnancy or around the time of birth. Lastly, dengue can be transmitted through infected blood, laboratory, or healthcare setting exposures. Dengue can be spread through blood transfusion, organ transplant, or through a needle stick injury. Now, let's look at the pathogenicity of the dengue virus. So, how does the virus actually behave once it enters the human body? The dengue virus attaches to the surface of a host cell and enters the cell by a process called endocytosis. Once deep inside the cell, the virus will fuse with the endosomal membrane and is released into the cytoplasm. The virus particle comes apart, releasing the viral genome. And then the viral RNA is translated into a single polypeptide that is cut into 10 proteins, and then the viral genome is replicated. Thusly, the virus assembly occurs on the surface of the endoplasmic reticulum when the structural proteins and newly synthesized RNA bud out from the ER. The immature viral particles are then transported through the trans-Golgi network, where they mature and convert to their infectious form. The mature viruses are then released from the cell and can go on to infect other cells.
Let's move to the impact of dengue to human. Dengue causes a wide spectrum of disease. This can range from subclinical disease to severe flu-like symptoms in those infected. Most people recover within a week or so. In some cases, symptoms worsen and can become life-threatening. Some people develop severe dengue, which can be any number of complications associated with hemorrhagic fever that affect organ system, severe bleeding, a sudden drop in blood pressure, organ impairment, or plasma leakage. Severe dengue have a higher risk of death when not managed appropriately. It happens when your blood vessels become damaged and leaky, and the number of platelets in your bloodstream drops. This can lead to shock, internal bleeding, organ failure, and even death. Dengue can affect all range of age, but nearly half of all dengue deaths are children under 9 years old. Children are particularly vulnerable to the disease because their immune system are weaker than adults, and they tend to play outside, where there is less protection against the mosquitoes. Dengue fever also can affect human emotion. As a human, when we're sick, we tend to feel maybe a little depressed and sad. This is because people are unable to go to work or do something important like usual due to this illness. So, the work will pile up or no income if they take medical leave. Besides, they feel less hungry than usual and lost their appetite. To suppress and eliminate viruses in our body, the human immune system activated. However, it costs a lot of energy that make the rising of the body temperature. So, if they lost their appetite, it will be leading to weight loss and other health and emotional issues. Dengue is a mosquito-borne viral infection which has been estimated to cause a global economic burden of $8.9 billion per year. 40% of this estimate was due to what are known as productivity costs, which is the cost associated with productivity loss from both paid and unpaid work that result from illness, treatment, or premature death. Direct costs represent over 70% of the total share for hospitalized cases. For outpatient, direct medical costs were low, but social costs were significant since indirect costs may account for up to 18% of the total cost.
So let's move to how government control dengue disease in Malaysia. There are three major components that Ministry of Health Malaysia focus on to control this disease. Vector control is the most effective method in preventing the transmission of the most devastating disease in the world, including dengue. Vector control program is a program to control the breeding of adult mosquito. These are some of the techniques that we use to control larvae. Surveillance or housing inspection can be done to survey larval presence, and it will undergo further identification. Not only that, daily monitoring on dengue cases and control activities also assist government to identify high-risk area and detect early outbreak. To control adult mosquito, fogging activities will be performed only when the disease is reported. This will help to kill any adult dengue mosquito that may be carrying the dengue virus. The insecticides used for fogging consists of numerous formulations with active ingredients. In fact, dengue vector control program in Malaysia rely heavily on these chemical controls. Not only that, government also had spread latest information regarding dengue disease through various platforms such as in social media, television advertisement, and radio. Through educating communities, early prevention activity can be done among them. A program funded by WHO implemented with MOH, COMBI, is a project-oriented approach that will objected to address the issue of dengue in the community and influence social norms toward a healthy behavior to make sure environment will kept clean. The enforcement of as stated outlines the Director General and Medical Officer of Health Authority to enter and inspect location, seize any items that may contain disease-carrying insect, order the destruction of disease-carrying insect, and conduct medical examination on people who may be infected with insect-borne disease. With the enforcement of this, the community become more sensitive and obedient to the authorities and able to avoid violation of the act as those that concern will be imposed.
Did you know that a pregnant woman already infected with dengue can pass the virus to her fetus during pregnancy or around the time of birth? Dengue can have harmful effects including death of the fetus, low birth weight, and premature birth. Therefore, to protect your pregnancy, if possible, avoid travel to areas with risk of dengue during pregnancy. Dengue is common in over 100 countries, including countries in the tropics. Find out if the country you will be visiting have dengue, and talk to your healthcare provider before traveling. Next, in a follow-up on dengue fever situation in Malaysia, in 2022, the Ministry of Health reports a total of 1,074 cases of dengue the week ending May 14, bringing the cumulative number of dengue cases reported to date to 14,725 cases. This number is up 45.2% compared to 10,139% in the same period in 2021. By state, Terengganu recorded the highest increase in the percentage of cases compared to the same period in 2021, with 164.72%, followed by Wilayah Persekutuan Labuan 150%, Kedah 137%, Negeri Sembilan 71.4%, Selangor 42.6%, Sabah 40.8%, and Kelantan
27.1%. Meanwhile, Perak 26.4%, Wilayah Persekutuan Kuala Lumpur 14.3%, Sarawak 30.7%, and Penang 3.7%. While in the states of Perlis, Melaka, Johor and Pahang recorded a decrease in their percentage of cases compared to 2021.